Good morning, everyone. My name is Jim Gardner. I'm one of the curators here at the Royal Tour Museum and welcoming you to our third last talk in our annual speaker series. This is on the eve of Burt Gummer Day. We won't be talking about graboids today, however. We'll be talking about poison arrow frogs and with our guest, Dr. Taran Grant. Taran is a full professor and associate curator of amphibians at the Museum of Zoology, both at the University of São Paulo in Brazil. He's an expat Canadian. He was born in Cranbrook, Vancouver, British Columbia, but has lived abroad since 1994 when he moved to Colombia to pursue his undergrad degree at the University of Bahia. And I apologize for my awful pronunciation of Spanish and Portuguese words. In 1998, he was awarded a joint fellowship at the American Museum of Natural History in New York and Center for Environmental Research and Conservation to pursue graduate studies in ecology and evolutionary biology at Columbia University. After finishing his PhD in 2005 and then a postdoc fellowship in computational biology at the American Museum of Natural History, in 2007, he joined the faculty at the Pontifical Catholic University of Rio Grande do Sul in Porto Alegre, Brazil. And then finally, in 2011, he moved to the University of São Paulo, where he's been ever since. And there, in collaboration with students and colleagues, he studies the evolution of amphibian diversity from a variety of perspectives, from the theory and practice of phylogenetic inference to more focused topics such as toad migration, the invasive biology of the American bullfrog, and the evolution of chemical defense. The world's richest countries in terms of amphibian species diversity are Brazil and Colombia, and most of Taran's work is centered in those countries. So today, Taran will talk about his research into chemical defense in poison frogs. Taran, I'll turn it over to you.
Thanks very much for the introduction. Thank you very much for the invitation to give a talk here. It's very strange to give a talk to my computer screen, so if I have moments where my mind wanders, that's the explanation. So yeah, the title of my talk today is Chemical Defense in Poison Frogs. Before getting into the talk itself, I'm going to do something I've never done before, but given that this is a talk to people in Canada, and I'm way in São Paulo in South America and Brazil, I figured I'd give a very brief sort of account of how I got here, because it's not the most usual place to be if you're a Canadian. So I was born in Cranbrook, and I spent the first 13 years of my life in Wausau. So I'm not sure if you can see my cursor, but there I am in Wausau. And then when I was 13, we moved to Vancouver. In fact, before that, we went on a trip. We went to Florida, and I discovered the amazing reptiles that are there. And so I got really turned on to reptiles. And then we moved to Vancouver, and I started volunteering at the Vancouver Aquarium, and that's where I got into frogs, and especially these poison frogs. This little green guy here is called Dendrobates auratus. And there I am with a wonderful mullet, looking very, very trendy. And when I was 15, I announced to my mom that I was going to go on a trip to South America. I was going to go see the Amazon. And so I gave her a hug goodbye and got my backpack and went on my trip. And didn't actually get to the Amazon on that trip, but I did go down as far as Ecuador and absolutely loved it. And I learned to speak Spanish. And then I moved back to Canada and tried to go to university for a while, but I was bored silly. I wanted to be in the tropics. And so I moved down to Cali in Colombia and did my undergraduate degree at the Universidad del Valle in Cali. And that's where I discovered that in addition to the brightly colored poison frogs, we also have all kinds of non-brightly colored and, in fact, not even poisonous poison frogs, which I'll get to that in a few minutes to explain what that means. But then it was after that, then I spent I guess a little over 4 years there getting my undergraduate degree. And then I moved up to New York to Columbia University and the American Museum, and then eventually down here to Brazil. So very, very briefly, that's sort of the trajectory that led me to where I am right now. Like I said, it's not the most usual thing for a kid from Cranbrook to end up in São Paulo. So with that out of the way, the general objective of my research on chemical defense in poison frogs is to test the generality, causes, and consequences of the variation that we observe in poison frog chemical defenses. And so that means that really there are two sides to this story, two parts to it. One is chemical defense, which is defined as the use of chemicals to reduce the risk of harm from pathogens, parasites, and predators. And this is an extension or an aspect, a component, I guess is a better word, of the innate immune system. And this chemical defense is often, at least in the cases that we're most interested in studying, it is tied to other behaviors and coloration and other aspects of life history that form these anti-predator mechanisms. And so that's where we have these frogs with warning coloration and interesting behaviors. And then the other side of this then is poison frogs. And so these here are some examples of the poison frogs. So this here is a Delphobates castanioticus. This one here is called Ranatomea amazonica. This is from the state of Pará here in Brazil. We've got Andinobates, this is the sister group of Ranatomea that's over in the Andes, so across the other side, a Transandean group. This is Phyllobates terribilis, pound for pound, or I guess gram for gram, one of the most toxic organisms on the planet. Also from Colombia, this one here. This is called Oophega histrionica. This would be one during the day, and here's a couple of them that are snuggled up beside each other, and we're sleeping under a leaf. They climb up into the leaves, maybe a meter and a half high, and they'll hide there and sleep at night because these are diurnal frogs. This is Oophega pomelio, called the strawberry poison frog. These are all really famous frogs. So even people that aren't into frogs like me know, or at least have seen these frogs before. This is called Dendrobates leucomelas, South American frogs. This is a Delphobates galactinotus. So these are really brightly colored, beautiful, very attractive frogs that attract a lot of attention from hobbyists and from scientists alike. This one here is called Amirga macero, which is from extreme western Amazonia. They have a unique kind of amplexus, which is the nuptial embrace that amphibians have. So the male grasps the female, but instead of actually holding onto her very tightly, he just sort of holds her gently, and he actually has some glands on the inside of his fingers that he'll rub against her. And what we believe he's doing is transferring pheromones to her. And so this is called cephalic amplexus, where the hands are sort of underneath the head like that. And then in this group of poison frogs, we also have tadpole transport. And so this is a male that is transporting a tadpole. And they will, in this group here, they will take them and deposit them in what are called phytotelmata. So in basically the axillas of bromeliads or holes in trees. So these are little water-filled cavities in vegetation. And so these are the poison frogs then. And they're the most famous of the frogs, at least or the amphibians when it comes to poison. But in fact, all amphibians have chemical defenses. And so we haven't necessarily tested all amphibians to see what the defenses are that they have, but they all have skin glands, and they have these granular glands, which is where the defensive chemicals are stored. They also have mucus glands, which I'm hoping you can see my cursors here. So this would be a mucus gland here where the M is, and here's a granular gland here where you can see all these little granules inside it. The D here is for a duct, and so these are dermal glands. And these ducts here then go up through the epidermis, and that's where the contents of these glands will be released. And so the granular glands are, as far as we know, exclusively for chemical defense, at least in the vast majority of cases. And these mucus glands then are for keeping the skin moist, for maintaining pH, for thermoregulation, for lubrication. They're used in reproduction and in parental care. And the reason that they have this system of chemical defense is that the skin of amphibians is effectively an external lung. So it's an extremely important organ for respiration. Some species of amphibians use their skin exclusively, or is the only organ that they use for respiration. So there are lungless salamanders. There's a lungless frog as well. There's at least one lungless caecilian as well. And the reason that this is so important is that a respiratory surface has to be very thin, has to be moist, You have to have the blood vessels very near the surface in order to have the gas exchanged, because the gases have to diffuse across this membrane. And so there is no way that a frog, or that amphibian in general, could invest evolutionarily in the kinds of mechanical defenses that we find in other tetrapods. And so in reptiles, we'll have scales, we can have feathers, we can have hairs, we can have bony plates, like in the turtles here. We can't have that in amphibians, because any sort of mechanical defense, at least a significant mechanical defense, would interfere with that ability to breathe across the skin. And so because of this, amphibians have evolutionarily in chemical defenses. And so we have a huge number of kinds of chemical defenses. This is a slightly outdated list here. I'm showing families here with different kinds of, with different classes of chemicals that have been found in those families in the skin. So we've got biogenic amines, we've got bufadienolides, lipophilic alkaloids, tetrodotoxins, pseudophrenamine, samandrines, peptides, and proteins. In the last few years, it's been found that there are microbial metabolites as well that are used for chemical defense by the amphibians, and so they'll have this microfauna on the skin, and these bacteria will produce substances which are used for defense by the frogs themselves or by the amphibians themselves. And we have terpenes as well that have been found in skin. So there's a lot of different kinds of chemicals here. And this is what leads to some of the most incredible diversity of amphibians that we see. So we've got cases of warning coloration here. This is a salamander from North America called Notothalamus viridescens. It has tetrodotoxin, which is a very powerful neurotoxin. Very few caecilians have actually had their skin chemistry worked out at all. But we see this warning coloration. And so even though these are fostorial organisms that most of the time, at least, we know that they live underground, they burrow underground, this coloration shows that they spend at least some amount of time on the surface where they'd be in contact with visual predators that would interpret this kind of coloration here as a warning coloration. There's another caecilian that's bright yellow. And then we also have frogs that have all this this kind of aposematic coloration. So this is a little tiny froglet here in the Atlantic forest of Brazil called Brachycephalus. This is Brachycephalus rotenbergi, and bright yellow here. This frog is only about 16 or 18 millimeters long. But in addition to these warning colorations, we also have warning behaviors that are really cool. And all this is part of an anti-predator mechanism. So even though all amphibians have these poison glands, in most of them, probably the poison is used mostly against pathogens. And then we've got these certain cases where the system is developed into an anti-predator mechanism, and then we can have this bright coloration. We can have these warning behaviors as well. So this is a frog from Asia that's called Bombina orientalis. And this movement that is doing, this position that it's in, is called the Unken reflex, where it raises. So dorsally, they are cryptically colored, so browns, greens, that kind of thing. They're camouflaged. And ventrally, then, they've got bright red or bright orange or bright yellow coloration. And then when they're threatened, they will raise up their hands and their feet and arch their backs, which is not an easy thing for a frog to do, and expose this bright coloration that they have ventrally. This is not exclusive to this group here. It was discovered in them first and described in them first, but it's also found in, for example, toads from South America here. This is a species of Melanophreniskis doing the same kind of behavior, and even in salamanders as well, where they'll do the same sort of what's called the Unken reflex. And here's another. This is Tarica. This is a genus of salamanders from North America as well. And then there's all kinds of other behaviors that we see that are associated with defense. And so salamanders will often exaggerate their glands, and they'll sweat sort of this milky substance all over their body, raise their tails and move their tails around so that they can wag them back and forth to call the attention of potential predators. Really pretty amazing behaviors that they'll do. often with this bright coloration. So here's salamander salamandra from Europe. This is an Asian salamander. So they'll often do this. They'll kind of lower their heads, close their eyes, and then, as I said before, raise the tails and sort of wag them back and forth and attract attention. Same thing here. We've got all kinds of really cool behaviors that these animals do that are associated with their chemical defense. And that's attracted a lot of attention of researchers over the years and of the public in general too. This is a toad, and they've got these big parotid glands on the side of their heads. And if you take those and give them a gentle squeeze, you don't want to do it too strongly, because you don't want to injure them. But if you give it a gentle little squeeze there, then the venom will shoot out of the poison will shoot out of the glands. This can happen. You can imagine being a predator and biting the frog or biting the toad and getting a mouthful of this poison. But it ends up that there are some species that can do this spontaneously as well. So this has been observed in this South American toad called Rabo guttatus, where they actually, the musculature of the pectoral girdle is kind of it functions such that they can apply pressure to that gland and they can shoot the venom out. And this has also been seen in this salamander as well, in salamander. So you might be wondering, if we've got all this, if all amphibians are quote unquote poisonous, or they all use chemical defense, and we've got all this diversity and these really cool behaviors and the bright coloration, what's so special about poison frogs? In fact, I forgot to mention this here. So beyond just all the cases that I showed before are of poisonous animals. Poisonous animals are those that do not have a mechanism of injecting or of delivering the defensive chemical into the predator. We also have a few species of amphibians that are actually venomous. And so these are salamanders that they are actually able to puncture their skin with their ribs. And so then if a predator is biting them, then they'll get a mouthful of ribs that have poked through these venom glands and would therefore be injecting the venom into them, like a snake or a scorpion. Here's another species that does that. And then, not to be left behind, we also have a group of frogs that does the same thing. So these are the casque-headed frogs here from South America. And you can see here where the spine is right at the edge of extends right to the edge of the skin here. And just to give you an idea of how spiny these guys are, pretty incredible skulls. And if a predator were to bite them in the head, or if they were to rub their head on a predator, then they would end up injecting a very strong poison that is actually, it's been shown to be more potent than pit viper venom. So these are highly defended organisms. But anyway, as I was saying before, if we've got all this diversity among amphibians, what's the deal with poison frogs? Why are they special? Why are they treated differently? And so they're characterized as being as possessing lipophilic alkaloids. So there's no exam at the end of this, so no one has to memorize any of this. But it's just one of the classes of compounds that are found in amphibians. To date, we've found a little over 1,400 different alkaloids that are referred to 26 structural classes. Poison frogs are all diurnal. They all have this bright warning coloration, and they are all microphagous. So they consume mostly ants and mites in their diet, and that's really important because the main characteristic that we have here with poison frogs is that they obtain their defensive chemicals not from their own genes or from their own, they don't produce them themselves, which is called biosynthesis, but instead from their diet. And so they obtain them exclusively from the mites and ants that are in their diet. Predominantly, it's mites. To date, over 100 alkaloids that are found in frogs have also been found in mites. And these represent 11 classes. So still a minority of the classes that have been found in arthropods. But we believe that at least the vast majority of these 1,400 alkaloids that we've found and 26 classes are found are derived from arthropods. And the technical term that we have for this is sequestration. So it's that this activity of consuming from some sort of an external exogenous source these defensive compounds and then incorporating them into the glands is called sequestration. So originally, the poison frogs were considered to be exclusively from this family called Dendrobatidae. In fact, all the frogs that I showed the first few slides, they were all from this family Dendrobatidae. But over time, as our knowledge has improved, we've discovered that that was an oversimplification, that we were dead wrong, and that actually this ability to sequester alkaloids from the diet and to develop this whole syndrome of characteristics, so that they're diurnal, they have this bright warning coloration, they use these lipophilic alkaloids that are obtained from their diet. That's evolved multiple times independently. And so in the next few slides that I'm going to show, the first number here, 96, is the number of species that are in that family that are poison frogs, so the ones that have those characteristics I mentioned before. And so 96 of the 205 species in the family Dendrobatidae are known or believed to be poison frogs. And in fact, even when you get into the family Dendrobatidae, it's not as simple as we once thought. So it ends up that the ability to sequester alkaloids has either evolved multiple times independently within the family, or it has evolved fewer times and has been lost independently multiple times. And the jury is still out on that. But what I've done here is I've labeled the different genus level clades in red, which are the ones that have alkaloids in them, that are technically poison frogs. So we have this common name of poison frogs, which is variably assigned to this family or to the frogs that have these characteristics, and which can then be part of this family and also be other families, which are the ones that I'll go through now. So the second group of poison frogs that was discovered is these ones here called Mantella. And there's about 265 species in this family, and 16 of them are in the genus Mantella, and they're the ones that have these alkaloids. They look externally very similar to Dendrobatides and Phyllobates, Adelphobates, these other poison frogs from South America, but they're from very far away, and phylogenetically they're very distant. And so the next one that was discovered is these ones here in the family Bufonidae. This is Melanophreniskis. So this is one that I showed a couple minutes ago. Interestingly, these 31 species are the sister group of all other Bufonids. And so we've got this big family of almost 650 species, and the sister group of all those other ones is this one here called Melanophreniskis, which they all have these alkaloids in their skin that they obtain from their diet. So they're all poison frogs. Then we have Myobatrachidae from Australia, and the most recently discovered one is a little group called the Limbatus group inside Eleutherodactylidae. So that's the phylogenetic distribution of them. Now to show you where these are found around the world, in Central and South America we have Dendrobatidae. Then we have Bufonidae, so this would just be Melanophreniskis, which is from the Atlantic forest region mostly of South America. We have Mantella over in Madagascar, Myobatrachidae, and explicitly Pseudophryne in Australia. And then we have this little group called the Limbatus group from Cuba. And so those are the poison frog groups that we know of, at least to date. There might be more out there, but these are the ones we've discovered so far. And my research, because I'm based in South America, is focused on these two groups here. And so that's what I'll talk about the rest of this talk here with the research that we're doing with these. So this species here, Olophaga pumilio, is commonly known as the strawberry poison frog, and it is without doubt the best studied and probably the most, among the poison frogs anyway, the most recognized poison frogs and one of the most recognized amphibians anywhere. It's been studied in great detail, in part because it's so bright and colorful and diurnal, and has really incredible behaviors as well, but especially because it is highly polymorphic. And so we have different localities, different populations that have really different coloration. And if we look at how those are distributed, what often happens is that we have some sort of an aquatic or other sort of barrier that are keeping these populations apart. And so then we have this local adaptation and the fixation of these different color morphs. And what's really cool about this is that we find in a lot of these cases that these local populations, which have a really unique coloration, also have a unique alkaloid profile. And so what I'm showing here Again, I'm hoping you can see my cursor, but each one of these little peaks here that's labeled is an alkaloid. It's a lipophilic alkaloid that was found in the skin of one of these frogs. And if we compare these different localities, we'll see that they have different profiles. And that's really interesting and has been a subject that's led to all kinds of research since the late 1960s. And so this is a case that is relatively well understood. It's the model organism of poison frogs. But so as I mentioned before, one of my aims is to test the generality of what we see in what we studied so far in the variation of poison in the defense of the poison frogs have. And so looking around here in South America, we have a very similar situation. We have this frog here called Adelphibates galactinotus, or called the splashback poison frog. And as you can see, it also has very different color morphs, and these are separated from each other geographically. So we look at a map here, we can see that each locality will have just one morph, one color morph. And so what I wanted to know is, do we have the same kind of pattern here in terms of the relationship between the coloration and the alkaloid profiles that we have in Central America with that Ophega familia, with the strawberry poison frog? And so we went to the field. We went to this locality here, where on opposite sides of this bay called Cachuana Bay, we have an orange color morph and a blue color morph. And what we wanted to know is, are their alkaloids different? So as I said, we traveled there. We got a plane, followed by a boat, followed by another boat, and then another boat, and arrived at the research station here. So this is me with a few of my students. And we went out into the forest and we found these frogs. And so here's a galactinotus that is in the leaf litter here. They're surprisingly hard to find, given how brightly colored they are. And then this here will be from the other side of the Cachuana Bay, where we got one of the blue ones. So it's sort of powder blue kind of coloration. So here's the localities that we sampled. So we took multiple specimens from each one of these localities. And then we looked at their alkaloids. And so from our study, we found a total of 89 alkaloids, representing 16 different classes. 72 of the alkaloids that we found are new for this species. 7 of the alkaloids are new for science. And if we compare to the only previous study that had looked at this species, they only had 10 alkaloids in their previous study, and we upped this to 72. And so here's just an example of one of these profiles, basically, of the alkaloids, showing just a few of them here. And we use nicotine here as an internal standard, so we can estimate the quantities, the absolute quantities of the alkaloids that are present in the skin. So nicotine is not a naturally occurring alkaloid in these frogs. If we then look at the distribution of these alkaloids across the orange and the blue frogs, we find that 46 of them are shared. 17 are unique to the orange and 26 are unique to the blue. If we look at this statistically, we find that is not significantly different. So the blues have a little bit more, but that's not even close to being statistically significant. And so this is a very different profile than what we find in the case of Permeable. So here we find that even though we have a very similar situation where we've got a barrier that's separating these two different color morphs, on either side of this barrier, the color morphs have the same alkaloid profile. So we don't know why that is. It might be because the arthropod availability is basically the same on both sides of this barrier, and maybe it's different in the Central American localities. There might be some other sort of an environmental characteristic which is affecting things. Maybe there's different predators in these two different places. That's difficult to imagine, but maybe that's what it is. Maybe there's differences in uptake efficiency in the case of the strawberry poison frog in Central America, and not in the case of the splashback poison frog in South America. This is an open question, and at this point, all we can really do is speculate as to why the situation for one species is one way, and for the other species it is another way. But that's the one of the purposes of the research that we're doing here is to try and figure out if what we see in one group is the same, if that's widespread and sort of characteristic of all poison frogs, or if it's unique. So another way of testing the generality of these patterns that we observe in dendrobatid poison frogs, is to look at other groups of poison frogs, which have been far less studied. And so here in South America, we have Melanophreniskis, which are these buffonid poison frogs. Their common name, actually, before they were known to be poison frogs, is red-bellied toads, and that's because they have these red bellies, like this. This is their distribution. So they're found in Brazil, Argentina, Bolivia, Paraguay, and Uruguay, mostly in the Atlantic forest, although they extend into the pampas as well, so into the grasslands as well. And so what I wanted to look at with this group, which had been, when we first started working with it, had very few studies looking at it, is we wanted to test and make sure that this really was a case of sequestration. We wanted to get a handle on variation, see if it varies in the same way that we see variation in poison frog, in denervated poison frogs, and then try and get a handle on, or some sort of an idea about what the cause of the variation might be. So first of all, sequestration, do they actually sequester, or is this different from the poison frog? So the way that we test this is by breeding frogs in captivity. So in this case here, it was a species called Melanophreniskis stelzneri, which was bred in captivity, and then we fed it alkaloids. In this case, we dusted fruit flies, and we fed the alkaloids to them. And so we have 3 different alkaloids that we're feeding to them in 2 different combinations. We have one that's called DHQ here, which is decahydroquinoline. And then we have 2 dye-substituted indolizidine alkaloids here. And we did this in different combinations. And that allowed us to make some interesting inferences. And so the first thing that we found here is that, yes, indeed, they do sequester alkaloids. And so in the lower panel here in B, we have the control frogs. So these are frogs that were raised in captivity, bred and raised in captivity. So they had never consumed alkaloids in their natural diet, which is basically fruit flies, is what we tend to feed them in captivity. And the ones up here in the panel A were the ones that were fed the alkaloid in their diets. We can see that they did indeed sequester this into their skin. And same with the other alkaloids. What was really interesting about this experiment that we did, though, is for the first time we were able to show that there was differential uptake efficiency. So what that means basically is that there's different, the rate of uptake or rate of sequestration is different for these different alkaloids. And so the decahydroquinoline was accumulated much more efficiently than the 5,8 dye-substituted indolizidine, and but it was sequestered more poorly than the 3,5 indolizidine. So we established that they do sequester just like poison dart frog, just like the denervated frogs do. So the next thing to look at then was the variation, and does it vary in the same way that we find variation in denervated frogs? And so here we have an example, or have some examples of the different coloration. In all these cases, they have red bellies, and so if they're they're darkly colored, or they're green, or they're sometimes they have some yellow dorsally, but it's always sort of camouflage, cryptic coloration dorsally, and then eventually they've got the red coloration. Here over on the left is the alkaloid quantity, and here are the species down below that we looked at. And so what we find is, comparing across different species, we have huge differences in the amount of alkaloid. And so this is all in micrograms. So admirabilis here has between 562 and 14,000 micrograms. So that is a ton of alkaloid there. This makes it them just as toxic as any of the denervated poison frogs would be. And then at the other extreme, we have here Melanophreniskis sammartini, which has several individuals that we looked at had no detectable amount of alkaloid, and some had as much as 6 micrograms. So very, very small amount of alkaloid there. And then if we look across in between, we see that it varies between these 2 extremes. And one of the things that you should call your attention here is how much it varies within species. And this is in the amount of, so this is between individuals of the same species. And this is in the amount of alkaloid, but then we say the same thing also with the richness, or with the number of alkaloids that we find. And so in admirabilis here, we have a total of 94 alkaloids that we found in that species. Here in sammartini, we have only 6 alkaloids that we found in the species. And then it varies in between there across. And if we look at how this varies among individuals, in admirabilis, it's anywhere from 37 to 57 alkaloids in the same individual. And in sammartini, at the other extreme, We have several individuals with no alkaloid and some individuals with up to 4 alkaloids, 4 different alkaloids in them. And then that varies in between here too. One of the things that we're concerned about is how dependent on sample size this might be. In some of these cases, we only have, usually we have 10 frogs. Sometimes we only have 5. But we have one species here that we collected 55 specimens. This was for a master's thesis. And so here we have a much better sample. And yet here we still only have between 5 and 17 alkaloids per individual, and only a total of 37 alkaloids in the whole species. So it seems like this is actually characteristic of these species. And the general pattern that we find then for variation is that most alkaloids are found in very few frogs, and few alkaloids are shared by most frogs. And this is the same sort of pattern that we find in other poison frogs. So in Permeable, for example, which is so well studied, and other dendrobated poison frogs as well, we see the same thing. So usually in a population, you'll have maybe 3 or 4 alkaloids that are found in pretty much everyone. So no matter how big your sample is, no matter how many frogs you collect, they all have that core group of alkaloids. And then as you sample more and more and more individuals, you basically have this great big huge tail in the distribution with 1 or 2 frogs that will have these different kinds, these additional alkaloids, so that they're shared by very few frogs. And so in this regard here, we're showing that the same pattern that we find in dendrobated frogs can be generalized over onto the melanophreniskis here. So whereas the Galactinotus had a very different pattern than what we see in Pumilio, for that hypothesis we were testing, in this case here, it's basically the same thing as what we find in dendrobated poison frogs. So it seems to really be a characteristic that's shared by all of them. So looking at the causes of this variation then, this is a central question in these studies of chemical defense. Why are defensive toxins so variable? And in the case of poison frogs, we've got a number of different sources of variation or causes of variation that have been identified in different studies. So one of them is this ability to sequester. So we think that is a genetically controlled characteristic of their physiology, basically, if they're able to sequester or not. So if you take a non-poison frog and give it alkaloids, it will not sequester the alkaloids into their skin. And within poison frogs, some frogs are able to sequester some classes of alkaloids and not others. And we showed already that there's different efficiency in the ability to sequester. So there's definitely a sequestration ability aspect of this, and we think that would be controlled by genes. But there's also variation across localities. There's variation over time in different seasons. Differences between sexes have been seen as well. So males and females having different alkaloid profiles, and that could be both the amount of alkaloid that they have and the richness of alkaloids, the number of alkaloids they have. Life history stages as well. Something else that causes variation would be biomodification. So it's been shown that in some poison frogs, they can consume one alkaloid and then convert it into another one themselves. Probably the main source, we think, of variation is their diet, because the alkaloids come from the mites and ants that these frogs are consuming. If you have a different diet from other frogs, then you will have a different alkaloid composition as well. That's probably the main one. And then another one here that's been identified is size. And so part of the problem with studying this, though, is that you have all kinds of different variables that you're looking at once. And so what I wanted to do is to look at the simplest case possible. So look at the same species. So we're missing out, or we're not looking at that interspecific comparison. We're removing that variable. Same species, the same locality, and the same time. And one of the hypotheses that had never been tested that would apply in this particular case is what's called the age hypothesis. And so the age hypothesis is pretty simple. You have frogs that when they complete metamorphosis, they have not yet consumed any arthropods that contain alkaloids, and so they don't have any alkaloids in their skin. And so they basically start off with 0, and then over time they will accumulate alkaloids so older frogs, you would expect, based on this hypothesis, would have greater richness and greater quantity of alkaloids. The thing is, though, when we look at a sample of these frogs, there is so much variation among individuals that pattern may not may not be real. So even though a priori we would think that this would be the case, there's good reason to think that maybe it's not. And so to test this hypothesis, what we have to know is the age of the individual. We have to know what the different defensive compounds are that those individuals have, and we need to look at that in both adults and juveniles. And then we have to look at the relationship between the defensive chemicals, the age, and size, and also we looked at the sex of these individuals. So we went to the field, up in the mountains here in Brazil here. This park is called Parque Nacional do Itatiaia, and it's there. This frog here, which is called the Moreri red-bellied toad, is the symbol of the park, because it's a charismatic little guy, and they're diurnal, so very popular. And we went there, and we chose this particular frog for this study for a couple of reasons. First of all, we'd already studied it, and we knew that it had alkaloids. It had these lipophilic alkaloids that it gets from the diet, but also there's an additional reason in their chemicals, which is that they have this compound here called bufotenin, and this is actually produced, it's synthesized by the toad. So they have simultaneously both these lipophilic alkaloids that they get from their diet, and also this defensive compound that they produce themselves, and that allows us to get at testing things in an interesting way. And then the other reason for targeting this toad specifically is that they undergo winter dormancy. And so even though we're in South America, and fairly close to the equator, in fact, in this case, this is way up high in the mountains. And in the winter, it can get as cold as -7 degrees, which I know for you guys, that's not really all that cold. In fact, I think today, Jim was saying that it was -5. So this isn't much colder than that. But for these little toads here, what that means is that in the winter, they'll burrow into the ground, or they'll use existing little holes in the ground, and they'll go into torpor. So they'll become dormant. And what this indicated to us is that there was a good chance they would have very clear, what are called lines of arrested growth, which are, if you take the long bones of these frogs, you do something called skeletochronology. So we do this with the femur. You can also do it with the phalanges. You can do it with other bones as well. But the femur tends to give the best results in our experience. It's just like taking a tree and looking at a cross section of it, and then counting the rings to determine how old that tree is. So in this case here, we've got the femur, which we took a cross section, and then we just count these rings here. So 1, 2, 3, 4, and there's experiences required to be able to interpret this properly, and there's a few little calculations that we have to do to make this work. But in general, that's the idea. It's the same idea as with a tree, counting the rings. And with this, we were able to determine the age of all the individuals that we captured. And so we had over 50 individuals. And so we had 7 juvenile females that their ages were between 1 and 5 years, 15 adult females that were between 3 and 6 years old, and 41 adult males that were between 2 and 6 years old. And we looked at the alkaloids that were present in the juveniles and the adults as well. And this is just to give you a quick overview of just a couple of examples that I pulled out. So here we have a 1-year-old juvenile that has only one alkaloid, this one right here. It's an allopomelia toxin. And they had no bufotenin yet. Then here we have a 3-year-old individual, which has 7 different alkaloids in it and has bufotenin here. Here's the peak for bufotenin. And then if we look at a 6-year-old, it has 13 alkaloids and also has bufotenin. And so obviously I chose these examples here to clearly illustrate the point that I want to make. But if we then look at this statistically, we find that because of our approach, we were able to tease apart the causes of the different, of richness and of the amount of alkaloids. So what we found here, this is a non-parametric multiple regression analysis. And what we see is that alkaloid richness is related to the age of the frogs. Alkaloid quantity, so the amount of alkaloid that is present, is related to the mass of the individual, or the mass of the skin, actually, is what we're weighing. And interestingly, the amount of bufotenin is also related to the mass of the skin, the mass of the individual there. And so this is kind of cool because we were able to tease apart these different characteristics of the alkaloid profile and attribute part of it to the age of the individuals and part of it to the size of the individuals. That doesn't exactly give us what the cause is, but it gives us a pretty good idea of what the cause might be. And so what we think is going on here, then we also found that there's no relationship to sex. And this is relevant because previously people had associated variation in alkaloids with sex. And what I think has probably happened in those other cases is that they didn't consider age as a characteristic that would explain this. And so females tend to be older than males. Something we can talk about afterwards if anyone has any questions about that. But so what we think is going on here with the richness and age relationship. So again, the older the individual, the greater the diversity or the greater the number of alkaloids that are present in the skin. What we think is happening here is that basically older individuals have been exposed to a greater diversity of arthropods. And so you can imagine being a very young toad, you've seen a couple of species of mites, and by the time you're 6 or 8 or 10 years old, you will have seen a lot more diversity of arthropods that you've consumed. And so if the alkaloids are distributed across different species of arthropods, then over time you would get a greater number of different kinds of alkaloids that are in your skin. And then as for the quantity and size relationship, so this is the amount of alkaloid and the size of the individual, what we think is probably going on is that this is a function of having greater storage capacity in large individuals. So this has not been looked at in these toads, but in other species of poison frogs, so in this case, it's the strawberry poison frog, it's been shown that there is a positive allometric relationship. What's a fancy way of saying that the number and size of granular glands increases with body size. So larger frogs will have larger, comparatively or proportionally larger glands and a greater number of glands than smaller frogs will, and that just means that they've got a greater capacity to for storage. And so that's what we think is going on to explain these two different aspects here and why we get different patterns. And then that takes me here to another question that we really can't get at from wild-caught animals, because that's what we're looking at before is wild-caught specimens. And that's this idea of differential uptake efficiency and this idea that there's something in their physiology that is determining how they're able to sequester. And so the way that we get at this has to be with captive bred individuals. And so we've got a breeding colony here of Amphibians, of Amphibians galactinotus, of the splashback poison frog. And so here you can see my student Adriana here. When she was my student, she's no longer my student, looking after the frogs for her project. And then here's the frogs eating Drosophila, so fruit flies, and that's the diet that we give them. So this is a source of food that does not have any alkaloid in it. And then of course here we have tadpoles that are growing. And the reason we have to do this again is because wild-caught individuals will have already consumed arthropods that contained alkaloids. And so if we want to study anything about how these guys are what the mechanism of sequestration would be, we have to breed them in captivity. And so in this particular study that I'm going to show to you here, we have our captive bred Amphibians galactinotus. And then what we're doing now, which is different from how we did this in years past, we're dissolving the alkaloid in ethanol, in a 50% solution of ethanol, and then we are injecting that straight into the mouth. So this is oral administration, and we did this with 2 different alkaloids. So we did this with synthetic decahydroquinoline and with a histrionica toxin. Again, there's no test as to what these names are, so these are just all you have to know is that these are just different classes of alkaloids here. And we had 4 different groups. We had a control group. We had a low amount group, which is they were administered 1 microgram of alkaloid per each feeding time or administration time. A medium amount, which is 3 micrograms, and then the maximum, which is 10 micrograms. And we administered the alkaloid to them for 14 days. And then to make sure that we were actually getting sequestration, so what was actually actively incorporated into the skin, and wasn't simply in the tissues and it was in the process of being cleared, we left the frogs for 7 days to clear out whatever was being metabolized and would be gotten rid of, so that we're sure that what we're actually seeing here is a result of sequestration and not simply a matter of not yet having cleared out the alkaloid from their system. And then we looked at the alkaloids that are present in the skin, in the liver, the kidney, and the muscle. And what we found, first of all, is that the alkaloid is present in all of the organs, but there's more in the skin. We also found that uptake is greater in the skin is greater for histrionic toxin than it is for decahydroquinoline, which is that goes again with this idea that we saw before in the previous study where there was differential uptake. So here we're showing that histrionic toxin is more efficiently taken up than decahydroquinoline. We also found that uptake in the liver was greater for decahydroquinoline than it was for histrionic toxin, which is interesting. And we found that the total quantity increases in the skin and liver, but that efficiency increases for the skin and decreases for liver. And so what we're seeing here is that's the slope of this line here, which is pointing down. As we increased up here with the skin, as we gave them larger doses of alkaloid, they sequestered a greater proportion of what we were giving them. So not only is the absolute amount of alkaloid that they're sequestering increasing, and does that in both cases, in this case here, the rate of sequestration increases when we give them higher doses. And that makes us think that there is, and then here in the liver, we see the opposite. And what we think is going on here is that at higher doses, for some reason, we know from other studies that we've done that the alkaloid is transported first into the liver, and then from there it is distributed out and will eventually reach the skin. And so what it looks like is that at higher doses, that is more efficient. And so that suggests that there is some as yet unknown mechanism, an enzyme or transporter or something like that is either activated or is increased at higher doses. And so that's why we would also have a decreased amount of alkaloid being left in the liver, because the liver isn't where these alkaloids are supposed to stay. They're supposed to be transported into the skin for defense. So that gives you sort of an idea of the research that we're doing here. I just want to close by a bit of a summary here, and hopefully, as I said a couple of times, there's no exam here, but hopefully what I've been able to do at least is convince you that chemical defense is really, really interesting and important for understanding the evolution of amphibians. It really sits at the center here of all kinds of different areas of evolution. So with studying phylogeny, we've got chemical defense in relation to the evolution of the chemicals themselves, the ability to sequester, or the compounds that the organisms themselves produce. We've got the evolution of these different kinds of coloration. We've got variation in morphology. Over with ecology, we've got predator-prey relationships, we've got resources. So in the case of poison frogs, where they're obtaining the alkaloids from their environment, that's a kind of resource ecology study. And then there's all these behavioral ecology that's related to this as well. And then for studying evolutionary processes in general, for natural selection, sexual selection, all this is sort of relevant to chemical defense. And so by focusing on chemical defense as a research area, you kind of it allows you to get into these other areas as well. And so finally, I just want to thank there's a lot of collaborators and friends and students and colleagues that I would love to be able to thank. My main collaborators on the poison frog or on the chemical defense studies are my colleague at John Carroll University in the US, Ralph Saporito, and then my two ex-students, Adriana Jackal, who is now at the University of Bern in Switzerland, and Patrick Colombo, who is at the Fundação Zoobotanica in Brazil. And so they were instrumental in doing this research. And thank you very much for your attention. If there's questions, I'd be happy to take them. And I will stop sharing now, I guess. There we go.
All right. Thank you, Taran. That was interesting. Certainly a lot more going on with chemical defense in amphibians than I realized. And as someone who sort of barely scraped through chemistry in university, I appreciate how you made the talk understandable for those of us who don't remember much about our undergrad chemistry.
Well, I should clarify too that I'm not a chemist, and so I associate myself with chemists to be able to do the research. So Ralph Saporito, my main colleague with this, he received his PhD's training at the National Institutes of Health with a guy named John Daly, who was the father of all this stuff, who published hundreds of papers looking at exactly this. And so I defer to Ralph on all things chemical, basically. So, and interesting,
good.
he's actually an ecologist too. So,
Ah.
but trained in chemistry. So,
right. No, so always good to have collaborators like that. So,
absolutely necessary. Yeah.
All right. So we have a few questions here.
Great.
I'm going to start with a couple from Ian McDonald. His first question is sequestration of poisons the same process as bioaccumulation of harmful chemicals in tissue, as in fish, for example, or is it a separate process?
That's a great question. I'm going to sort of hedge a little bit and say we don't know because we don't actually know what the mechanism of sequestration of alkaloids is. And so that's one of the active things that a lot of the different research groups that are looking at this, we're all sort of trying to get after that. We think that it's different because we think that it's mediated by some sort of enzyme. So we think that at some level it's active. Whereas in the case of bioaccumulation of heavy metals or that kind of thing, it's passive, right? It accumulates, unfortunately, naturally. In this case here, because we can feed the alkaloids to some species that are incapable of sequestering and to other ones that are capable of sequestering, we think there's some sort of an active mechanism that's involved in this. So they are probably different mechanisms. But again, because we actually don't know what the mechanism of sequestration is, I can't give a definitive answer.
Fair enough. Thanks. Okay, Ian's second question is to ask how common sequestration-derived toxins are versus those that are derived in other ways.
So very uncommon. So of the, what are we at now? About 8,500 species of amphibians, more or less. There are maybe 300 species approximately. That's a bit of an overestimate, actually. Let's stick with that. 300 species, just to be generous, that are able to sequester alkaloids. So in all the other cases, it appears to be that the frogs produce their defensive compounds themselves. There are a couple of exceptions to that. One in particular that I'm gonna draw attention to is the case of tetrodotoxin, where we don't really know what the source of that is. So this is the same compound that is present in the pufferfish. And so for sushi, there's the delicacy of the pufferfish liver, and you have to be careful with eating it and all the rest of it, right? So in that case, it appears to be a bacterial symbiont that is the source of the tetrodotoxin. In the case of amphibians, it's a little more complicated. And in part, it's because of the incredible amount of tetrodotoxin that some of these species have. So that salamander that I showed before, the one that was arching its back, the Tarica granulosa, there are populations of that salamander that contain in a single salamander, which maybe this big, enough tetrodotoxin to kill 40 people, and a huge amount of tetrodotoxin there. And the amount of tetrodotoxin that bacteria produce is so tiny that in order to generate that much tetrodotoxin, you'd have to have a stadium full of bacteria, basically. And there's also some curious things in salamanders, especially in that species, which over time the amount of tetrodotoxin they have varies, and it doesn't vary in a way that seems to be consistent with it being bacterial in origin. So there are some of these classes of alcohol or classes of compounds that we really don't know what the source is, but the vast majority is biosynthesis by the organism themselves, and most of them are undoubtedly not for an anti-predator function. So in the majority of cases, it's probably antimicrobial peptides, that kind of thing, that are that they serve to kill pathogens, basically.
Interesting. Okay, we have several questions from, and I apologize if I mispronounce the name, Karthik. Obviously very interested in this subject. So their first question is, would chemical defenses be an intrinsic character to all this amphibians? And is there any reason to suggest this may have applied to ancestralless amphibians such as temnospondyls, especially the smaller ones.
Yeah. I would put money. I mean, this is a question that's you could probably answer better than I could, Jim. But I would put money on this being something that was present in temnospondyls. We have no way of testing it, unfortunately. Mm-hmm. It's kind of like that projectile tongue. I would put money on that also being something that evolved far earlier. Given they're in the same habitats, my prediction would be that it evolved way before the Amphibia.
Right. Yeah, it's a very interesting question. I too have no ideas about how one might test that.
Time machine. Get going with the time machine, Jim.
All right, next question from Karthik. Are chemical defenses in frogs symplecomorphy, or are selection pressures strong enough to cause the independent origins of each type of toxin? And are some toxins or taxa united by the composition of their toxins?
So that's also an excellent question. And not the easiest one in the world to answer because our information is so spotty. And so definitely we have clades that are characterized by certain classes of defensive chemicals. So, we've got these genera of poison frogs, for example, and this is definitely the ability to sequester alkaloids has evolved independently in these different genera. So there's a synapomorphy there. What's interesting to see is the rarity of different classes of defensive compounds co-occurring. So, for example, there is no instance, to our knowledge, of an amphibian that has both lipophilic alkaloids and tetrodotoxin, even though within Dendrobatidae, we have some species that have tetrodotoxin. So one is a water-soluble, we can call it an alkaloid. There's a ring with a nitrogen there, so we can call it an alkaloid. That's tetrodotoxin. And then we've got the lipophilic or the fat-soluble alkaloids, and they do not co-occur. And same thing over in the bufonids. So we've got a Telopus, for example, which they've got tetrodotoxin. They do not have lipophilic alkaloids. And so same thing with antimicrobial peptides, for example. It's very rare to find any kind of evidence of antimicrobial peptides occurring with anything else. So we do have some cases of biogenic amines occurring with peptides, things like that, but it's pretty rare. So I think that, it also comes down with how we're defining characters, right? But if I had to stake out a position right now, which I kind of do because the question came, I would say that yes, chemical defense is going to be, with this, what we have now would be a synapomorphy of Amphibia. And then within that, there will be different classes of defensive compounds that will have arisen independently. And exactly what the order, the sequence of that would be at this point, I have no idea because the information is too spotty.
Great. All right. How are poison frogs not affected by the toxins they take in from their diets? And is there a coevolutionary relationship between poison frogs and their prey?
So there's no coevolutionary relationship between their prey and the poison frogs, to our knowledge. There is a great deal of research looking at how they are resistant. This is the technical term for this is autoresistance. There is some evidence indicating or suggesting that this would be mutations to ion transport channels that would render those channels immune or resistant to these different compounds. This is quite well established in the case of tetrodotoxins, not poison frogs, but it's a much better studied system. So I'll start with that. In the case of tetrodotoxin, we know which channel it affects. So it's the sodium channel. And we know, in fact, it's domain number 4, which is where the active site is. And then we can also see mutations that occur there in organisms that are resistant to TTX or tetrodotoxin. So that's a really cool kind of starting point. The problem that we have with poison frogs is that there are over 1,400 different alkaloids in 26 different classes. And so it's a lot more difficult for us to figure out what the mutations might be for a specific one of these alkaloids, especially because most of them, we don't even know what their biological activity is. And so in sort of broad strokes, we have some ideas, but looking at it in detail, it's very confusing. And so a few years ago, there was a paper published that identified mutations in an ion transport channel that were purported to confer resistance against batrachotoxin. And then a couple of years later, that was disputed. And so this is absolutely an active area of research that lots of us are looking at. And that was the first half of the question. What was the second half? Can you repeat it, please, Jen?
Nope, because I already deleted it.
Well, there you go.
But
my answer was too long anyway.
Karthik has a few more.
Good.
Yeah. So how has the
oh, sorry. No, I already had answered the second question, which was if there's a coevolutionary relationship between the predators and the prey.
Right.
There you go.
Yeah. Okay. Good. Yeah. So how has the chytrid disease that's ravaging some frog populations does that affect toxin production and metabolism?
So interesting. So focusing on poison frogs, which is the group that I know best. The reason that it's called batrachotoxin dendrobatitis is because it was that BD was first discovered in captive specimens of Dendrobates tinctorius. So it can definitely infect poison frogs. Interestingly, poison frogs do not seem to be declining. So non-poison frog Dendrobatids have been decimated. And so what used to be this big, now we know it's polyphyletic, this group that we used to refer to as Cholestethus, which now is Hyloxalis and Alabates and Cholestethus, which is a smaller version of it. A lot of those species are gone. So the first species that I discovered, which are now in Ectopoglossus and Hyloxalis, those have been extinct since the mid 1990s, and were almost certainly wiped out by BD. And in fact, in Central America, the first indicator that BD is starting to wipe through a population is some of these species, what are called rocket frogs, the old Cholestethus, these non-poisonous poison frogs. So these are dendrobated frogs that don't have alkaloids. They start showing up on the forest floor as being as dead, or swabbing them and finding that they've got BD. So against BD, or it could be something behavioral. It's been shown in other taxa, so in toads and especially in different groups of true frogs, that the antimicrobial peptides that they produce can make them resistant to BD infection. So chemical defense is definitely related to this.
So some are lucky and some are not,
depending on what they're producing. Probably
right.
the most affected group in Central and South America would be Atelopus, which is these buffonid frogs that have tetrodotoxin. And even though they can have large amounts of tetrodotoxin or TTX analogs, and can be quite poisonous, evidently BD has no problem with wiping them out. So unlucky.
Right. Okay. The last question we're going to ask, because we're crouching into the lunch hour here. Another one from Karthik. Does the lethal dose of the toxin released by a frog act as a proxy to the size of the largest predator in the range?
That's a very complex question to answer. Let me see how to attack that question. We don't know hardly any of the potential predators of poison frogs. So there are examples of we know that the visually oriented predators are the ones that will be seeing these visual signals, and so we assume that birds are potential predators. We also know that there are some snakes, or Liophis, that can consume poison frogs. But for the most part, it seems like these ones that at least that are quite poisonous probably don't have any predators. So, or at least if they do, they're very few. So, for example, if you go to something, and this is based on the abundance and the boldness of these frogs, and so you can go to some of these localities that I've been to in the Choco in Western Colombia, and it's just sort of this. As you're walking along, there can be this sort of this yellow wave of frogs that are kind of going along the edge of the stream as you walk, and your shadow is cast across them, and they're really, really abundant. And if there were a predator that could come down and consume them, they'd have a heyday. So, the other thing that makes this difficult, and this is a problem in the way that studies of poison frogs have been approached in the past, is that, in fact, the reason that we refer to this as chemical defense as opposed to poisons, when we're talking about this technically, is that whether something is poisonous or not is subjective, right? It depends on what the predator is, and so something that is poisonous to to Homo sapiens may not be poisonous to a snake or to a bird, right? Or to an arthropod. And we know that, for example, that spiders can attack these frogs. And also these alkaloids don't occur in isolation. So when, for example, when John Daly would take these alkaloids and would inject them into a mouse to determine what the toxicity is, he's testing it against, the standard organism, which is the mouse, which is not a natural predator of these frogs. And also he's doing it one alkaloid at a time. And in fact, what we have is these cocktails of alkaloids. So a relationship to the size, the amount of alkaloid, the amount of defensive compounds that are in a given frog, does not appear to be related to the size of the potential predator. And part of the reason that I say that is because within a species, we don't have a whole lot of variation in the size of the frogs, but we can have a huge variation in the amount of the alkaloid that's present. And so one of the examples that I gave there was that green frog called Melanophreniskis admirabilis, where we had, I think it's 500 micrograms up to almost, I think it was 14,000 micrograms of, so 14 grams, right? Well, is that right? Yeah, 14,000 micrograms of alkaloid. And so within basically the same size organism has a hugely different amount of alkaloid present.
Yeah. Oh, very interesting stuff. And I really want to thank you for this talk. Really quite illuminating and opened my eyes. I don't think we'll look at Kermit quite the same way.
Well, thank you very much, Jim. Thanks very much for inviting me to give this talk. I should say that I went to the museum and met you in July, I guess it was.
Yeah,
and
last year.
I was impressed beyond words with the museum. It was fantastic. I became a huge fan of it. And so it's a real thrill for me to be able to talk with you. So I would have preferred to do it in person. It's like I said before, it's a little weird to be giving a talk to your screen like this. You can never see if it's going over well, or if you're going too long, or if everyone's bored. But it's been a lot of fun. So thank you.
Yeah, you're very welcome. All right. And before we sign off, I'd like to invite all you cat aficionados to attend next Thursday's talk, which will be on the history of cats. Okay, so we'll see you next Thursday at 11 Alberta time. Thanks. Have a great week.
Thanks, everyone.